Hello guys, hello friends, hello G's, it's your boy again, Falling with a Star. So guys, today our topic is gluconeogenesis, which other people also call it neogluconeogenesis. Now my first question is, what is gluconeogenesis? It is the synthesis of glucose from non-carbohydrate molecules, for example, pyruvates, lactates, glucogenic amino acid, TCA cycle intermediates, glycerol, and also propanol-CoA. So these are some of the examples of what molecules that can be used to synthesize glucose through what gluconeogenesis. Gluconeogenesis is not a single separate pathway, but a combination of what glycolysis, TCA cycle, and also four key reactions of what gluconeogenesis. So all these come together to form what the gluconeogenic what pathway. Now my next question is, where does it occur? It actually occurs both in the cytosol and in the mitochondria, both in the cytosol and what the mitochondria. Part of the gluconeogenic pathway occur in the cytosol, part also occur in the what in the mitochondria, and it also occurs in what organs like the liver, the kidney, and also the small intestine. The liver, the kidney, and the small intestine. Now my next question is, when does it occur? When does it occur? It actually occurs during the fasting or starvation state, after glycogen stored in the liver is being depleted. You see, during the starvation and the fasting state, your body will first rely on the stored glycogen for glucose. You understand? You see, glucose is being stored in the liver in the form of glycogen, and during the starvation state, this will be the first target for your body to break it down by the enzyme glycogen phosphorylase to get the glucose. After the glycogen stored is being depleted, now your body will use another way to get glucose, and that pathway is what we call the gluconeogenesis. There are certain organs that solely depends on glucose for energy. Organs like the brain, the neurons, the erythrocytes, the islands, for example the cornea, the testes, and also the renal medulla. These organs solely depend on glucose for energy. Now my next question is, what is the importance of gluconeogenesis? It actually help clear metabolic products like lactates, glycerol, and propanol-CoA from the body. You see, lactate is being produced in the muscle during anaerobic glycolysis, and this lactate can be harmful to the body. It can cause lactic acidosis. So gluconeogenesis help clear this lactate, because this lactate will be used to form glucose through the gluconeogenic pathway. Glycerol, also during the starvation state, triacyl glycerol is being broken down to glycerol and fatty acid, and that glycerol is being used in gluconeogenic pathway. In that case, we actually clear this metabolic product. And also propanol-CoA during beta oxidation of all chain fatty acids. We get this propanol-CoA, and we can use it in gluconeogenic pathway. So in so doing, the body the body is able to clear this metabolic product. Glucose is also important for maintaining intermediates in the TCA cycle. You see, in glycolysis, glucose is being converted to pyruvate, which will be converted to acetyl-CoA, and acetyl-CoA will be used in the TCA cycle. So acetyl-CoA will be converted to the TCA intermediates, and in so doing, glucose helps to maintain these intermediates. Now, the next thing I want us to discuss is the similarities between glycolysis and gluconeogenesis. You see, gluconeogenesis is the reverse of glycolysis, but not the exact reverse. Glycolysis actually involves 10 steps with 3 irreversible steps, and these steps are known as the bypass steps in gluconeogenesis. And now the question is, what are these 3 steps? You see, the step 10 in glycolysis is the conversion of phosphoenol pyruvate to pyruvate by the enzyme pyruvate kinase, and this step is irreversible. So this step is being bypassed in gluconeogenesis. Also, fructose phosphate to fructose 1,6-base phosphate, the step 3 in glycolysis, is irreversible by the help of the enzyme phosphofructokinase 1. So in gluconeogenesis, we bypass this step. Also, glucose is being converted to glucose phosphate in step 1 in glycolysis, and this step sorry, this step is also being bypassed in what gluconeogenesis. So these are the bypass steps, the 3 bypass steps in what gluconeogenesis. So in gluconeogenesis, we are going to use another way. In gluconeogenesis, this is our aim. We are going to use another way to convert the phosphoenol pyruvate. No, it is the reverse. So we are going to use another way to convert the pyruvate back to phosphoenol pyruvate, because we cannot use this enzyme. We are going to use another way to convert fructose 1,6-bisphosphate to fructose 6-phosphate by another enzyme, because we cannot use this enzyme. We are also going to find another way to convert glucose 6-phosphate to glucose by another enzyme, because we cannot also use this enzyme called hexokinase. So guys, let's look at how to actually get glucose from pyruvate. We all know that in glycolysis, the end product is pyruvate, and this pyruvate is in the cytosol, because glycolysis occurs in the cytosol. So this pyruvate will get into the mitochondria. And when it gets into the mitochondria, it is being acted upon by a certain enzyme called pyruvate carboxylase. This enzyme actually requires a coenzyme called biotin. This pyruvate carboxylase will convert pyruvate, which is a 3-carbon compound, to oxaloacetate, which is a 4-carbon compound, meaning that carbon has been gained in the form of carbon dioxide. And also in this step, energy is also being needed in the form of ATP. This oxaloacetate would like to come out from the mitochondria into the cytosol, but this is the case that it doesn't have the ability to pass through the membrane of the mitochondria. So this is what it will do. It will convert itself to malate first, and this malate has the ability to pass through the membrane of the mitochondria. So the malate will come out from the mitochondria into the cytosol. When the malate gets into the cytosol, now the question is, what happened next? This malate will convert itself back to the oxaloacetate by the help of the enzyme malate dehydrogenase. So this oxaloacetate in the cytosol will now be acted upon by a certain enzyme called the PEP carboxykinase, phosphoenol pyruvate carboxykinase. And this enzyme will actually convert the oxaloacetate into phosphoenol pyruvate. Phosphoenol pyruvate. And this reaction actually needs energy in the form of GTP. So we use energy in this step. So this phosphoenol pyruvate will follow the glycolytic pathway, but in the reverse form. It will follow till the time it gets to fructose 1,6-bisphosphate. And we said that the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate is irreversible. So we cannot use the enzyme phosphofructokinase 1 to convert the fructose 1,6-bisphosphate back to the fructose 6-phosphate. So we need another enzyme. So this step is a bypass step. So when it gets to this part, there is this enzyme we called fructose 1,6-bisphosphate. This is the enzyme that is actually going to help convert the fructose 1,6-bisphosphate back to fructose 6-phosphate. And this fructose 6-phosphate will be acted upon by this enzyme we called phosphoglucoisomerase. It will be converted to glucose 6-phosphate. But listen to this. We said that the conversion of glucose to glucose 6-phosphate is irreversible. So that means in gluconeogenesis, this step is bypassed. So we cannot use hexokinase to convert glucose 6-phosphate back to glucose. So we need another enzyme, and this enzyme is being found in the endoplasmic reticulum. So this enzyme is called glucose 6-phosphatase. So this enzyme is the one going to help convert the glucose 6-phosphate back to glucose. And this glucose, remember that this is actually occurring mainly in the liver. So after getting the glucose in the liver, it is being pushed through a channel we call GLU2, glucose transporter 2, into the blood, and in so doing we replenish the blood glucose level. So this is how we get glucose from pyruvate. You understand? So let's look at how to get glucose from other non-carbohydrate substances. So guys, let's look at how we can get glucose from lactate. And this reaction is what we call the Cori cycle. So this is the muscle. And in the muscle, glucose is being converted to pyruvate. And during anaerobic condition, the pyruvate is being converted to a lactate. And the muscle will push this lactate into the blood, and the blood will transport the lactate into the liver. And in the liver, the lactate is being used to form what? Glucose. So lactate will be converted back to the pyruvate, and the pyruvate through gluconeogenic pathway will be converted to what? The glucose. And this glucose will be pushed back into the muscle to be used in glycolysis for what? Energy. And this cycle is what we call the Cori cycle. Let's look at how to get glucose from what? Alanine. And this is what we call the glucose-alanine cycle. Also, in the muscle, the glucose is being converted to pyruvate. And during transamination, this pyruvate is being converted to what? Alanine in the muscle. And the muscle will push this alanine into the blood will transport it into the liver. And in the liver, the alanine will undergo another transamination back into the pyruvate. And the pyruvate will follow the gluconogenic pathway for us to get what? Glucose. And this glucose will be pushed from the liver back into the what? The muscle to be used in glycolysis for energy. And this is what we call the glucose-alanine cycle. So now let's look at how to actually get glucose from what? Glycerol. During starvation, triacylglycerol is being broken down into what? Glycerol and what? Fatty acid. This glycerol will be acted upon by an enzyme we call glycerol kinase to convert it to what? Glycerol 3-phosphate. And this glycerol 3-phosphate has the ability to be converted to a glycolytic intermediate we call dihydroxyacetone phosphate by the help of the enzyme glycerol 3-phosphate dehydrogenase. Glycerol 3-phosphate dehydrogenase. And this dihydroxyacetone phosphate will actually follow what? The glycolytic. It's a glycolytic intermediate, so it can follow what? The gluconogenic pathway for us to get what? Glucose. And this is how we get what? Glucose from what? Glycerol. We can also get glucose from what? Propanol-CoA. This propanol-CoA will be converted to succinyl-CoA, and succinyl-CoA will be converted to what? Malate to oxaloacetate, which will be used in what? In the gluconogenic pathway for us to get what? Glucose. Also, let's look at this. How to get glucose from what? The gluconogenic what? Amino acids. You see, this is the gluconogenic or glucogenic what? Amino acids. This amino acid, glycine, alanine, cysteine, serine, threonine, tryptophan, can undergo transamination to convert themselves to what? Pyruvate. And the pyruvate will be used in the gluconogenic pathway to get what? For us to get what? Glucose. The same way, aspartic acid can undergo transamination and convert itself to what? Oxaloacetate. And this oxaloacetate can be used in the gluconogenic pathway for us to get what? Glucose. The same applies to this glucogenic amino acids, glutamate, arginine, histidine, proline. They can undergo transamination into what? Alpha ketoglutarate, and this also be used for us to get what? Glucose. The same way, phenylalanine, tyrosine will undergo transamination to form what? Fumarate, and this also follow the glucogenic pathway for us to get what? Glucose. The same applies to valine, isoleucine, and methionine. So that's it. So now let's look at the regulation of what? Gluconeogenesis. Gluconeogenesis is actually active in fasting and starvation period. And we have some 4 key enzymes for what? This gluconogenic what? Pathway. And they are the pyruvate carboxylase, PEP carboxykinase, fructose 1,6 bisphosphatase, and also glucosase phosphatase. But when it comes to regulation, these 2 enzymes are the ones being regulated: pyruvate carboxylase and also fructose 1,6 bisphosphatase. And this regulation can either be allosteric regulation, hormonal regulation, or covalent modification. So let's look at something here. During starvation, I said that triacylglycerol is being broken down into what? Glycerol and what? Fatty acid. And these fatty acids can undergo beta oxidation to produce more of what? Acetyl-CoA. So it will lead to what? An increase in what? The level of acetyl-CoA. And increase in acetyl-CoA can actually activate pyruvate carboxylase, which is a key enzyme what? In gluconeogenesis. And also inhibits what? Pyruvate dehydrogenase, which is a key enzyme in what? In TCA cycle. Also, we have this substrate called fructose 2,6 bisphosphatase, actually being formed by an enzyme called phosphofructokinase 2. Phosphofructokinase 2. And this high level of this fructose 2,6 bisphosphatase will actually inhibit a key enzyme in the gluconeogenic pathway called fructose 1,6 bisphosphatase, and also activate phosphofructokinase 1, which is the key enzyme in what? Glycolysis. But the opposite of this will do the reverse. You understand? The opposite of this will do what? The reverse. Also, glucagon, hormonal regulation. Glucagon causes phosphorylation of enzymes And phosphorylation of enzyme in gluconeogenic pathway will actually activate them to help in the glucogenic pathway. And also insulin will do the opposite, so dephosphorylation. So insulin will actually dephosphorylate the gluconogenic enzymes to actually inhibit them. You understand? So they are fighting each other. You understand? Also hormones like ACTH, growth hormone, glucocorticoids, all of these hormones help increase gluconeogenesis. They help increase gluconeogenesis. So guys, if you haven't subscribed to this channel, please subscribe to this channel for me. And I have videos on anatomy, biochemistry, and physiology. Stay safe. Bye.